This is a nerve tissue histology tutorial presented by the SchrDbb. We reference Atlas of Histology with Function and Clinical Relation by Dongmei Cui. So here we're going to see the nervous system. Generally, the nervous system is involved with impulse transmission from one part of the body to another in order to maintain homeopoiesis or hemostasis, in order to maintain hemopoiesis, in order to maintain homeostasis. So we need to know that word. So we are going to visualize a neuron and the synapses. So we have said that the neuron is involved with the transfer of action potentials because they are channels there are different channels and at the level of the neurons which are involved with that. Now we have also synapses. We need to know, we are going to visualize the histologic section of synapses later. Now we need to know that generally the nervous system can be divided based on its structure and also be divided based on its function. So the division of central nervous system based on the structure is going to be divided into central nervous system and peripheral nervous system. Central nervous system is a part of the nervous system which is involved only with the brain and the spinal cord, while peripheral nervous system are the spinal nerves and the cranial nerves. Those are the peripheral nervous system. Now the division with respect to function is central nervous system based on division based on the function is divided into two. We have the voluntary or the somatic nervous system and we have the autonomic nervous system. Now we need to know that there are two main types of cells inside the central nervous system. We have the nerve cells and we have the Glial cells. The Glial cells are cells that do not originate from the central nervous system, but they are present there in order to give additional function to the nerve cells, while the nerve cells are the cells that originate from the central nervous system, originate from the nervous system, and then they function in the nervous system. So now let's start with the neuron and synapses. Now we need to visualize that this is generally the schematic representation of a neuron. So the neuron, this is an example of a multipolar neuron. It is made up of a cell body, and that cell body is called the soma. Then we have a nucleus here. Then we have dendrites. So the dendrites are usually small processes which are connected to the cell body, while the axon is a longer cell process. So when you have it's a single long cell process, that's the axon. So we have an axon here, and then you have myelin overlying the axon. And usually in the peripheral nervous system, myelin is produced by the Schwann cell, while in the central nervous system, myelin is produced by the oligodendrocytes. And then this is the axonal terminal. Now, and at the level of the axonal terminal, we have the synapses. Now let's list some types of neurons. Now there are three main types of neurons. You have unipolar neurons, you have bipolar neurons, and you have multipolar neurons. The unipolar neurons, you need to know that the cell body is not in contact with the axon. So we have a stalk or a cell process that is connecting the cell body with the axon. So that's it. So the axon is not in contact with the cell body, here there is a stalk or a cell process that's connecting it with the axon, and this is usually specific for the case of somatic sensory nerves. So somatic sensory nerves have this usually. Now the second one, we have the bipolar neurons. Bipolar neurons are mostly in specialized somatic sensory. This is mostly in generalized somatic sensory, like the sensory nerves involved with touch, vibration in the body. While this is specialized somatic sensory, like the sensory nerve for vision, the sensory nerve for olfaction, that is smelling, the sensory nerve for hearing is bipolar. So the bipolar neuron, here we have a long dendrite and then you have also a long axon. So the axon is in contact with the cell body. So a long dendrite and axon are the different terminals of the neuron. Why now a multipolar? So we have a single dendrite and single neuron, and then this is the dendrite terminal, this is the axon terminal. Single dendrite, single axon in case of a bipolar neuron. Now the next one is a multipolar neuron. So multipolar neuron, you have the cell body which is connected to multiple dendrites, and then the axon, there is a single axon with the axon terminal. We also have the axon collateral with its terminal. So those are the different types of neurons. So after visualizing the types of neurons, you are going to have the different neuronal cells. So these types of neurons here are mostly involved with the peripheral nervous system. So in peripheral nervous system, we mostly have this, and we also have this at the level of the tracts. But now in the central nervous system, the nerve cells, we have both the neurons. The neuron are one type of nerve cells, and we have other nerve cells here. So these are the other neurons, other nerve cells. So this nerve cell is called the Purkinje nerve cell, and the Purkinje nerve cells are found usually in the cerebellar cortex. They're found usually in the cerebellar cortex. You see how they have multiple dendrites, as you can see here, and a single cell body. We have number 2, we have stellate cells. So stellate cells are these cells here. So you see here's a central cell body, and then we have radiation of the dendrite, as you can see here. Then we have pyramidal cells. Pyramidal cells are usually located in the cerebellar cortex, and they usually span the length of the cerebellar cortex from layer 1 to the layer 6. The next one, we have the spinal motor cells. It's just like they resemble the Purkinje cells. We have the central, and then you have radiations of the dendrites. Yeah, this is the image, and then the next one is the unipolar cells. And unipolar cells are usually located in the generalized sensory, with the dorsal root ganglion located at the level of the spinal cord. Though these are unipolar cells, they have the same diagram just as this. Now, the next thing to visualize now are the stains of the nervous tissue. Now, you need to know that we have different stains in the nervous tissue. The first thing, we have the Golgi preparation stains. The second, we have the Niels stain. The third, we have the myelin stain. And the fourth is hematoxylin and urea stain. Now, the Golgi preparation stain is used in order to visualize the processes. So when you want to visualize the dendrites, the axon, you use the Golgi preparation stain. And they can be used in visualizing the Purkinje cells, just can be visualizing the cerebellar cortex as here. The second is a Niels stain. So the Niels stain is mostly going to visualize the rough endoplasmic reticulum. That's why it's going to help you to visualize the cell body, as you can see here. So if you want to see the cell size, the cell bodies, if you want to locate the cell bodies, you can use the Niels stains. The next one is the myelin stain. The myelin stain is used to stain the myelin sheath. So when it stains the myelin sheath dark, as you can see this. That's why the white, the white matter, as you can see here, is stained dark because the myelin sheath stained myelin process darker. Now the next one is hematoxylin and eosin stain. The hematoxylin and eosin stains can stain all the neuronal cells, but you see the distinction is not quite alright. You can just tell you on the cell body, you can visualize the stains often all the myelin cells involved also with pathologic condition, and sometimes normal neuronal to stain also normal neuronal cells, but mostly in pathologic condition when you want to visualize necrosis and all that. Now, this now is the transmission of nerve impulses. Now, if you have multiple dendrites here, so these are the presynaptic neurons, and this is the postsynaptic neuron. So the presynaptic neuron are going to send impulses to the postsynaptic neuron. The postsynaptic neuron is going to send impulses later. This now brings us to a synapse. So what is the schematic representation of a synapse? This is a schematic representation of a synapse. We have this presynaptic membrane with the synaptic knob. Here you have the microtubules, you have the axon terminal with the axon button. So terminal button or the synaptic knob. So this is a mitochondria, these are the synaptic vesicles. At the postsynaptic membrane now, we have the postsynaptic density with the mitochondrion and then the dendrite. So it's located at a particular dendrite here. Now, this is the electron microscopic view of a synapse. So the membrane of the axon. So this is the membrane of the axon here. These are the mitochondria. This is the mitochondria here. These are the synaptic vesicles. This is the synaptic layer. Now this is the postsynaptic membrane with the postsynaptic membrane. You see the postsynaptic with the postsynaptic density, as you can see here. So this was the postsynaptic density, postsynaptic membrane. This is the presynaptic membrane, the synaptic vesicles. So in order for you to distinguish histologically the presynaptic from postsynaptic membrane, you can see the synaptic vesicles in the presynaptic membrane, and then the postsynaptic membrane, you're going to see the postsynaptic density histologically. So that's what you can visualize. Now, then this is another structure of a mitochondrion. Now, so this is the dendrite here. So here is the dendrite, here is the axon terminal, and here is the axon terminal. Here is the dendrite, here is the axon terminal, here is the axon terminal. So you can see also the presynaptic and the postsynaptic membranes here. So this is the synaptic zone, the axon terminal, the round synaptic vesicles. You can see the vesicles here. So usually the axon terminal are going to be the end of one nerve. So this is a presynaptic membrane, this is a postsynaptic membrane, the presynaptic membrane, postsynaptic membrane. So you can see the postsynaptic density here. Then here are the round synaptic vesicles. It's similar here. Here is the mitochondrion, here is the synaptic density, here are the synaptic vesicles, postsynaptic vesicles here, flattened vesicles here, and then the axon terminal. Now, this diagram is going to just show you the gross anatomy of the central nervous system. So you need to know that what on the embryologic origin of the central nervous system, the central nervous system is going to be divided into three main parts. We have the fore, we have the midbrain, and then we have the hindbrain. The forebrain is called embryologically called the prosencephalon, and the prosencephalon is going to produce the telencephalon later in life. Now the midbrain is going to be called and the prosencephalon, the prosencephalon is going to divide into two. It's going to produce the cerebral hemisphere called the telencephalon, and it's also going to produce the thalamus and the structures which are close to the thalamus called the diencephalon. So all this produced by the forebrain, also called the prosencephalon. Now the midbrain is going to be produced from the mesencephalon, and it grows to produce the midbrain, still called the mesencephalon in later life. Now the brainstem now is called the rhabdoencephalon. So now the rhabdoencephalon now is going to divide into two, called the metencephalon and the myelencephalon. The metencephalon is going to produce both the pons and the cerebellum, and the myelencephalon is going to produce the medulla. Then you have the spinal cord. Now the transverse section of the spinal cord is going to now show so it's going to show the gray matter of the neuronal cells. It's going to show the posterior horn, show the intermediate horn, the anterior horn. All this is the gray matter, and laterally you have the white matter with the columns. So this is the myelinated axon. So now at the end now of the white matter, we have now the two roots. So we have this is the spinal nerve here. So the spinal nerve, before it enters the spinal column, the spinal cord is going to divide into two. It's going to produce the posterior spinal rod, and then it's going to produce the anterior spinal rod. The posterior spinal rod is going to transmit the generalized somatic nerve, And then in the posterior spinal root, you have, since the generalized somatic nerve are unipolar neurons, they are going to have their cell body out of the central nervous system, and that cell body is going to lie in the posterior root, the posterior dorsal ganglion. Now, also, why the motor neurons usually are multipolar, and since they are multipolar, they have very small dendrite, and then their cell body are going to lie in the gray matter of the central of the spinal cord. Now, we need to also know that there are also ganglions which are going to be produced by the autonomic nervous system, usually sympathetic nervous system, because sympathetic nervous system have presynaptic nerves, and then there are postsynaptic nerves. The presynaptic nerve and postsynaptic nerve are going to produce the cell bodies of the postsynaptic nerves are going to lie also inside the ganglion, also a central nervous system called the synaptic chain ganglion. Now I'm going to show you histologically later the difference, how you can make histologically the difference between the unipolar sensory neuron or the posterior root ganglion histologically, and then the sympathetic chain ganglion histologically. Now, the next thing to visualize now is going to be the cross section of the peripheral nerve. Generally, you need to know that the cross section of peripheral nerve, just like the muscles, the peripheral nerve, one peripheral nerve is going to be covered by the endoneuron. One nerve is going to be covered by the endoneuron. Now, there are multiple nerves which can be moved in a fascicle, just like a motor. Multiple nerves can pass through a fascicle, and that fascicle is going to be covered by the perineum, and then lastly, covering all the nerve bulk is the epineuron. Now, this thing that has been used here is a Toridine blue stain. So Toridine blue stain is the one that is in the instead, and then this stain here is the one which is trichome stain. Okay. Now, the next one now is a posterior root ganglion. So how is the posterior root ganglion? These are the elements found in the posterior root ganglion. You can see axons, you can see unipolar neurons, you can see satellite cells inside the posterior root ganglion, and you can see blood vessels. So the posterior root ganglion is an enlargement of the posterior peripheral nerve root of the spinal cord, as I've already told you. It is made up of unipolar sensory neuron, cell bodies, and also their axons. It is going to be made up of satellite cells. It can also be made up of Schwann cells. Schwann cells, which is involved with the myelinations of the periphery, the somatic sensory neurons, can also be present there. So all this, and the main glial cell is going to be located there. You have Schwann cells, and then you have satellite cells. Satellite cell is going to be just like the astrocytes, in the central nervous system, where the function here in order to give nutrients to the central nervous system, the Schwann cells. They give nutrients to the central nervous system, to the nerve of the peripheral nervous system. So now, the clinical correlation. So the clinical correlation of this disorder is going to be the hereditary sensory motor neuropathy type 3. So hereditary sensory motor neuropathy type 3. This is a profound loss of large myelinated axons shown in this is a hereditary demyelinating neuropathy. So it's a hereditary demyelinating neuropathy is best associated when compare the density of the normal cells and then the abnormal cells. As you can see here, we have normal. Here is stain. You see the stain that has been used here is Toridine blue. So you see this stain here is compared with this. So here you have a high myelination. So in order for you to visualize myelination, you can either use myelin stain, you can use Toridine blue stain to visualize myelination. So here you have highly blue, so it is well. But here it's going to be demyelinated. The blue color has changed. So it is highly demyelinated in hereditary sensory motor neuropathy type 3. Now, myelinations and the myelinated and unmyelinated cell. What is the schematic representation? So this is how a myelinated cell is represented on the peripheral nervous system. So you have a central neuron, and then you have circular processes cytoplasm of the Schwann cell. So the Schwann cell is like this. Also, this is another diagram. So we have circular process of Schwann cell. This shows in case of of a myelinated neuron. But the myelinated neuron, you have the Schwann cell is still having a cell membrane that is going to cover the neurons, but the neurons do not have those circular processes just like here. Now you see, you have a Schwann cell with a cell membrane covering each of them, but it does not have circular processes containing myelin inside. Is it clear? So here it is unmyelinated axon. Now this is a myelinated peripheral nerve on longitudinal section with a trichome stain. So when you use the trichome stain here, you can visualize here you have another stain that can also be used in case of to visualize myelin is a trichome, and that this trichome is, you see, is dark color just like what you have seen above. So here you visualize here the nucleus. This is a schematic representation of the nucleus. You have the Schwann cell, you have the knot of Ranvier. But here on the histologic section, you also see it. You have the axon, you have the knot of Ranvier, you have the cleft of the Schmitz lantern, and the cleft of the Schmitz lantern are actually protrusions, are actually in contain cytoplasm. The cytoplasm, those cytoplasm provide metabolic support for the membrane of the myelin coating. So just like this, you see this coating like that, this membrane coating like that, turning like that is the cleft of the cleft of Schmitz lantern. Now, multiple sclerosis, unlike the hereditary disorder that you have seen above, here in multiple sclerosis is instead an autoimmune inflammatory demyelinating disease. So now here you have usually occurring in young women between 20 to 40 years. Now here what happens is that the histologic section, where you're still going to have demyelination when you use toluidine blue, but here we have used a Luxol fasting. Here you are going to see plaques of degenerated myelin, as you can see here. So we have plaques of degenerated myelin and a part of normal myelin and all that. Now the next thing here we have so this diagram shows an electron microscopic view in the longitudinal section of the myelinated neuron. So this is the knot of Ranvier here. These are paranodal cytoplasm, and this is the myelin sheath. So the paranodal cytoplasm, you can still call it what? The cleft of Schmitz lantern. So those paranodal cytoplasm are the cytoplasm of the Schwann cells, as you can see. So this is that. So we have exposed axon membrane. Now the next one we have here, this is the major dense line. So this is a myelinated neuron. Now this one has been taken on transverse section, this one was longitudinal section. This is a myelinated neuron. Myelinated neuron, why? Because you can visualize the nucleus of the Schwann cell, and then here we have the myelin sheath. The myelin sheath, you have myelin sheath that can be seen here. This is the axon in the inside part here. There are multiple microtubules, as you can see, and these are the circular processes of the cytoplasm of the Schwann cell. Now in contrast, this one is unmyelinated because you have a central nucleus of the Schwann cell. This is the membrane of the Schwann cell, and then you have multiple unmyelinated axons at the periphery of the Schwann cell cytoplasm. Now the next thing to visualize is the peripheral sensory receptors. So there are multiple peripheral sensory receptors, just like the example we have the Merkel cells, you have the muscle spleen, you have the Merkel cells, you have the nerve endings, and you have the Messier corpuscle and the Pascal corpuscle. But the only one that can easily be seen on light microscopy are going to be the Messier and the Pascal corpuscles. So this is the schematic representation of a Messier corpuscles that has a capsule. You visualize a capsule above. And it is beneath the epidermis of the skin, usually the epidermis of the palms of the hand. So we have a capsule above, and then this is a Messier corpuscle. Why for the Pascal you have a capsule, and then you have myelin sheath with nerve. Here the nerves are entwisted, as you can see here, while the nerve is just straight with the capsule. So the Messier corpuscle, so this is a histological representation. So we have a capsule here. So you have the nerve ending. Actually, it is a bit trying to intertwine, as you can see here. While for the Pascal corpuscle, we have a capsule, and in that capsule it's not more intertwining. So you have instead road zones, and you have a core. So here it is like a more and more of a white space, and here it's more like intertwining. And then the capsule is thicker in case of Pascal corpuscle, while the capsule is less thicker. And then the location of the Messier corpus is located directly underneath the epidermis in the dermis. So it's directly underneath the epidermis, and here the Pascal corpuscle is usually in the hypodermis. Now the spinal cord. You have used here the myelin stain. So here you can visualize now the white matter and the gray matter. So when you visualize the white and the gray matter, here you see the columns. These are the part of the gray matter here are going to be columns, while the part of the white matter, the part of the gray matter is going to be the cord, the horns, while the part of the white matter are going to be the columns. So you can visualize the posterior column, the anterior column, the lateral column here. The posterior column, you have the fascicle gracilis, and you have the fascicle coniatus. Now, the posterior column, fascicle gracilis, fascicle coniatus. You have the lateral column constituting all the corticospinal tract. Then you have the gray matter with the horns. You have the anterior horn, the intermediate horn, and then the posterior horn here. Now this is a spinal cord imaging now with histologic section with Neal stain. So with the Neal stain, you have the capillary, you have neuron cell body, you have axons, you have glial cells here. So you can visualize glial cell nuclei. And then you need to know that what the glial cells are cells that do not originate from the central nervous system, but they play a role in the central nervous system. These are glial cells, these are the neuron cell bodies with the Neal stain. These are some axons. Axons can easily be visualized with the Neal stain. So this part here is more of white matter because it does not have many cell bodies, and this part here with more neuronal cell bodies is going to be the gray matter. Now you need to know examples of glial cells. So examples of glial cells, we have the oligodendrocytes. Example of so we have oligodendrocyte, which is involved with myelination of neurons in the central nervous system. We have the second one, we have astrocytes. Astrocyte is involved with the nutrition of the cells in the central nervous system. We have the microglial cell, which is involved with macrophaging. The cell are involved with phagocytosine in the central nervous system. We have other cells. So other cells in addition we are going to see later. So the next one now, this one is a neuron in a reticular formation of the brainstem. So these are how the neurons are going to resemble. Now this is the cerebral cortex. Generally you need to know that what the cerebral cortex has different layers. It's divided into layer 4, into layer 1 to layer 6, and those parts of the cerebral cortex have different cells which are lining it. Usually you need to know that what in layer 1 it consists predominantly of horizontal running dendrite and axons. So in layer 1 consists of horizontal running dendrite and axons. Layer 2, this layer 2 and layer 4 in the microglia usually consists of small, tightly packed neurons. It consists of small, tightly packed neurons. Now, in layer 3 here and layer 5 consists of larger neurons. So this ones here is smaller. So this is horizontally packed neurons. This is tight. So this is horizontal neurons. So type 1. Type 1 consists usually of horizontal running dendrites. This one is small, tightly packed neurons with this one, and this one is large neurons. So these are the different characteristics that you need to point out. Now, in the cerebral cortex, you need to know that the major cell involved in the cerebral cortex is the pyramidal cell. And now this stain here, which has been used in the pyramidal cell, is a garbage preparation stain. You can see A is the axon of the pyramidal cell. So we have the axon, you have the apical dendrites here, which is going to protrude through the layer 1. This is layer 6 here. So the pyramidal cell protrudes, moves from layer 1 to layer 6. So here is the basal dendrites, which is located close to the soma or the cell body. So the cell body is usually located at the level of the layer 5, because we said that there was large dendrite protrusion. This is located in layer 5. This is in the layers above, and this is the layer below. Now, the next thing to visualize here is Alzheimer's disease. So Alzheimer's disease is the major cause of dementia. So it's a common form of dementia. This is much more of it's a common cause of dementia in elderly. So it's a progressive. So characteristic is that you have a progressive memory loss, changes in cognitive and personality changes. Now, what is the main disorder with Alzheimer's disease? So it is due to the deposition of the A beta proteins in intracellular neurofibrillary tangles. But the problem here is that these proteins are usually amyloid bodies. So that is a major problem. And now on the histologic section of this, you're going to see amyloid plaques. This is the normal cell here. How it's going to resemble under a Belshovsky stain. So we have the normal. This is normal cell, and this cell here is the one which is having amyloid bodies present. This dark stain with amyloid plaques. Now the next one, you have the cerebellar followed with the new stain. So this is how the cerebellum is going to resemble. The cerebellum also has its white matter, and it also has a gray matter, just like the spinal cord. So it has a white matter and the gray matter. So you need to know that when the central, generally when you are in the brain, the white matter is located in the center, while the gray matter is located at the periphery. But when you are in the spinal cord, the white matter is going to be located at the periphery, and the gray matter is going to be located at the center. So since you're in the cerebellum, also in the brain, we have the gray matter at the periphery and the white matter at the center. So usually at the end of the gray matter, we have of the cerebellum, we are going to have the cerebellar cortex, and then the cells usually which make up the cerebellar cortex are usually the Purkinje cells in the cerebellum. Basically. Now the next one, this is still the cerebellar cortex. You have just taken this and you have magnified this section. So this is still the cerebellar cortex with Purkinje cells. We have and granular cells. So we have the Purkinje cells with the Purkinje cell bodies. So this is that you have granular cells that you can also visualize. Now the next one is encephalocele. Encephalocele now is a clinical correlation involved with the cerebellum. Since the cerebellum is the main one which protrudes through the abnormal orifice in the brain, so in the head, so in the skull. That's why it is involved with this clinical correlation. So here, when you have, so this is the abnormal protrusion of the brain with the myelin. So abnormal protrusion of the brain with or without the myelin through an orifice, through an orifice in the skull due to abnormal formation of the skull bone. So that's an encephalocele. Now on the histologic section, you are going to have here the problem is that the skull has not been produced, so the skin is directly in contact either with the meninges or within the nervous tissue, that is the brain. Now the next one we have the meninges. Now the meninges, for the meninges you need to know that the meninges are divided usually into three. The dura mater, you have the arachnoid mater, and then pia mater. So the arachnoid mater has the arachnoid barrier here. It has a subarachnoid space. In the subarachnoid space, the arachnoid trabeculum, as you can see here, or the arachnoid web, and then you have the blood vessels in the subarachnoid space, and then you have the pia mater with the nervous tissue underneath. Now this was a schematic representation. This is now a histologic section of that. So the dura mater can be seen. You have the epidural space, which is closer to the bone above, and then you have the arachnoid wall, so the arachnoid barrier just underneath the epidural mater. Now this is the subarachnoid space. In the subarachnoid space now, you can see now blood vessels inside. And then since this has been taken from the spinal cord, this is the posterior root of the spinal nerve, and then here close to the spinal cord you have the pia mater. So the pia mater is the one that overlying the spinal cord. Now the clinical correlation is meningitis. So for meningitis, it's just the inflammatory disease of the meninges. Full stop. That's meningitis. Now the inflammatory disease of the meninges is mostly of infectious origin, but we may have other forms of meningitis, so which may be also due to autoimmune irritation of the meninges. So meningitis, inflammatory disease of the meninges, mostly of infectious origin. Now you need to know on the histologic view of meningitis, usually the diagnosis is made with a lumbar puncture and CSF analysis, but on the histologic view of meningitis, you will see inflammatory cells. Normally, this was the diagram seen here. You see this space normally was very clear, but here you see it's very dark because we have in the arachnoid space is filled with inflammatory cells and proteinaceous fluid. This one is much more dark compared to this diagram here, which is clear. Now the next thing to visualize are going to be the types of the glial cells. I've said that the glial cells are all the cells that do not originate from the central nervous system but play a function in it. An example of a glial cell, you have astrocytes. Astrocytes are involved with giving nutrition to the nerve cells. Then we have the astrocytes produce, when they are in contact with the blood vessels, they have a part called the glial limitans, and that glial limitans, the major part which is involved with the blood-brain barrier. Now we have microglial cells which are involved with phagocytosis, bacteria inside the central nervous system. Just play a role. It's just derived from macrophages. Then oligodendrocyte is involved with myelination of cells inside the central nervous system. And then you have other glial cells. Example, the radial glial cells and the ependymal glial cells. Now astrocytes. So this is just a geographic population of astrocytes that you can visualize here. So this is the protoplasmic astrocyte. Then these are some pyramidal cells here. So the astrocytes are going to give nutrition to these cells. Now the clinical correlation of astrocytes now is a glioblastoma, and a glioblastoma is a form of astrocytoma. It is a highly malignant tumor that arises in neoplastic astrocytes. So how do you visualize glioblastoma on the histologic section? So you visualize it with abnormal capillaries 1, you visualize it with the live tumor cells 2, you visualize with the necrotic tissue 3, and you visualize with zone of palacidin. Now the zone of palacidin is a configuration which occur because of so we have zone of necrosis and non-necrotic tumor cells that align themselves in a parallel array. So when you have a zone of necrosis, so like this place here, which is white here, a zone of necrosis, while this other zone here is zone of tumor cells. So the zone of necrosis and the zone of tumor cells, which are lying in a parallel array, is going to be called the parasitic. So it's going to be called the parasitic zone. Now, autonomic nervous system. So this is generally autonomic nervous system divided into sympathetic and parasympathetic nervous system. The sympathetic nervous system is going to be thoracolumbar, while parasympathetic nervous system is going to be craniosacral. So these are the cranial nerves. The major nerves which are involved in parasympathetic nervous system are cranial nerve 3, cranial nerve 7, cranial glossopharyngeal nerve, and then the vagus nerve. Those are the major cranial nerves. Then you have the sacral nerves S2 to S4, which are involved with parasympathetic nervous system. Then you need to know that both the sympathetic and parasympathetic nervous system need to produce ganglion before they arrive the target organ. So these are the different ganglion, the ganglia involved. So for cranial nerve 3, you have the ciliary ganglion is going to supply to the eye. For the cranial nerve 7, you're going to have the first ganglion, which is the pterygoid ganglion, is going to supply the lacrimal gland. The pterygoid ganglion is going to supply the lacrimal gland, and the particular is going to supply the lacrimal gland in the eye. And then you have the submandibular ganglion is going to supply the submandibular gland, the sublingual gland in the mouth, and then you have the otic ganglion. Otic ganglion comes from the glossopharyngeal nerve, otic ganglion, to supply the parotid ganglion in the oral mucosa. Now we have the next one, the vagus nerve. You need to know that the vagus nerve supply everything in the viscera. You supply the lungs, we supply the heart, we supply the stomach, the duodenum, the pancreas, the liver, the intestine. So it's going to supply all the bladder. So this is going to supply the intestine. And then you need to know that the vagus nerve stops its supply at the level of the splenic flexure, or the distal two-thirds of the transverse column. So it stops there. Now what continues is the sacral nerve, and the sacral nerve continues to supply from there downwards. So downwards you supply the pelvis, that is going to supply the bladder, is going to supply the bladder, is going to supply the genitals, even the kidneys. So now for the sympathetic nerves, you need to know that it's thoracolumbar. You start from T1 and end at the level of L2. So it's going to supply now. It's going to first have all the nerves. Usually all the nerves of the sympathetic nerve are going to have their ganglia in the sympathetic chain. So usually cervical ganglia and all the sympathetic chain ganglia here, except three ganglia which do not have their origin in the sympathetic chain. So this ganglia, the ciliary ganglion, which is going to supply the foregut. We have the superior mesenteric ganglion, which supply the midgut, and then you have the inferior mesenteric ganglion, which is going to supply the hindgut. Also, you may have the aorticorenal ganglion, which supply the hindgut. So those are the different elements that you're going to see here. Now, you have already seen the posterior spinal root ganglion. Now you're going to see now the sympathetic ganglion. How does it resemble? In this posterior spinal root ganglion, we have spoken about satellite cells. We're talking about the posterior spinal root nervous neurons, cell bodies, and then we have spoken about their axons. But here you still have satellite cells which are located here, and satellite cells are involved in nutrition of the cells here. Then you have the sympathetic motor neurons. You see, this one is actually a bit quite different from the other one. So you have sympathetic motor neuron, you have axons, cytoplasm, you see the nucleus, see the nucleus. Now the next one here is the myenteric plexus. So the myenteric plexus and the Auerbach plexus. The myenteric plexus is here, just between the circular smooth muscle and the longitudinal smooth muscle, while the Auerbach plexus is beneath the longitudinal smooth muscles. Now, submucosal plexus. The submucosal plexus is just located inferior. So this is the Meissner plexus. So submucosal plexus or Meissner just inferior to the mucosa. So this is submucosal plexus. Now we need to now see the comparison between the posterior root ganglia and the autonomic ganglia. Based on the cell body arrangement, the posterior root ganglia is going to be arranged into groups within the ganglion. Cell bodies are variable in size. So we have groups within the ganglion, and then the cell bodies are varying in size. So here in autonomic, they are going to be evenly distributed within the ganglion and uniform in size. Here they are variable in size. Now the cell body characteristic. The cell body characteristic in posterior root ganglia is going to be round cell body and central nucleus. While here we have multipolar cell body and we have eccentric nucleus. The eccentric nucleus is at the center. Now for the satellite cells inside the posterior root ganglia, we have complete capsules of the satellite cells inside posterior root ganglia. Now we have incomplete capsules of satellite cells in autonomic ganglion. And lastly, we have synapses in the ganglion. You have no synapse in the posterior root ganglion, and you have numerous synapses in the autonomic ganglion. So for you, thanks for your kind attention. Don't forget to like and subscribe for your channel. Thank you.